Welcome to Health Professional Radio. I'm your host, Neil Howard. Thank you so much for joining us for another segment. We're going to be speaking with Brian Dugan this morning. He's joining us here from QIAGEN to discuss the more than 3 billion biological samples tested Milestone and the more than 1 million samples with its next generation sequencing or NGS panels. Welcome to Health Professional Radio. Brian, how are you?
Thanks for having me, Neil, and great to be with you.
Well, give us a little bit of insight into your professional background and tell us a little bit about what QIAGEN is and what your role is there.
Yeah, well, thanks for having me. And so to start off, my name is Brian Dugan. I'm the Senior Director of Next Generation Sequencing, Global Product Management, and Head of Enterprise Genomics. At QIAGEN, my role, I've been with QIAGEN since 2015. Moving into different marketing and product management roles. I have a background in a master's in science of biochemistry from Georgetown University. QIAGEN itself, right, is one of the largest providers of consumables and reagents in the life sciences tools market. And so, QIAGEN today provides products for the isolation of nucleic acids such as DNA and RNA. It provides assays for PCR, right, for testing, which many people may know now because of COVID testing, getting sort of more reliable testing through PCR. We have products like our digital PCR platforms, and we also have our next generation sequencing portfolio, which I'm lucky to be a part of.
Now, I understand that you've, as I suggested, have reached a couple of significant milestones as far as biological samples tested and samples tested with these NGS panels.
This year, we passed the 3 billion sample mark for our sample preparation technologies, right? So when I speak about that, I'm talking to you before you do any test, any molecular test, that's PCR, next-gen sequencing, a specific gene test. It requires you to isolate DNA or, like I said, RNA from a sample. And so, QIAGEN has been in business now for the last few decades, and in that time, we have produced, market-leading kits. And so, from our samples, I think we can say at the front end of almost all molecular testing downstream, and we've been able to, pass that 3 billion mark, which is really, I think, incredible, and it shows the trust that consumers have in our products in using them. And many of the times, they're in things like working with COVID testing, obviously cancer testing and other types of things that are used downstream, not to mention all the research applications that we support with our isolation products.
Now, what does this this particular milestone mean for your company? You mentioned that, it's evidence that your clients trust you, your products, your developments. What does it mean for your company overall and for the customers, aside from the trust that they're demonstrating?
Yeah, so I think what it means QIAGEN is obviously a leader in the space and has been for a while. I think it just continues to underpin, the need or our desire to continually push the envelope and to innovate. We've innovated, I think, in many ways with the sample isolation technologies. We're going into, areas like liquid biopsies, which are on the cutting edge. But also, as we talk about next-gen sequencing, we mentioned the fact that with our QIAseq panels, we passed 1 million samples of next-generation sequencing use, in just 5 years. And so what we see is the market has evolved, and we continue as a company to evolve and to meet the needs and push into new areas so that we can, essentially, service our customers both in life sciences, early development, and all throughout the spectrum of testing.
For our listeners who aren't familiar, what is NGS or next-gen sequencing, and why is it so important, especially now?
Yeah, that's a great question. So next-generation sequencing is really, I think, an evolution of molecular testing over the last 20, 30 years, right? So if you go back and you see what next-generation sequencing is, a technology that's, you can call it massively multiplex sequencing. And what that allows you to do is look at billions of bases at the same time from the genome or from a sample. And you can look at it at a genome level, a transcriptome level, which is all the RNA. But it's really a technology where you can take that sample, you can prepare it so that it works on a sequencer, and that sequencer allows you to see all the individual bases in the genome. And then you take that information and you compare it against, say, a reference genome, and you look at that and say, wow, this person may have a mutation in a gene. That gene could be inherited disease. It could be for a cancer gene. It could be something. And we've seen where next generation sequencing 10 years ago, it hadn't really penetrated into the clinic. But now we see that there are many companies out there, clinical companies, running large next generation sequencing panels out there, delivering results to customers. And while I work on the life sciences side, right, with research customers, in the majority of my work, we see that this is completely changed, right, testing over the last decade of time.
Now, as far as assays associated with NGS, how do they work?
So an assay is you take that nucleic acid, that DNA and RNA, you do what's called library preparation. It can be either a general library preparation where you look at, say, the whole genome, or it can be a focused or targeted application where you really enrich for certain areas. Like a cancer panel, you may look at, a collection of, a handful or hundreds of cancer genes at the same time, and you get that ready. The library is prepared. You put it on the sequencer, and then, as I said, it spits out a file. That file is called a FASTQ. But really, that file has all the genomic, that individual base information. And when you do the comparison and that analysis, that allows you to potentially deliver a report or look at data that says, oh, this person, this case, this sample, this person, right, in clinical cases, it may have a mutation, it may have something important, or it may not, right? But it allows you to look at things and go through and see potentially up to the whole genome.
So what would you say is coming next for NGS overall, and where does QIAGEN fall into play there?
Yeah, it's interesting. I go back to, I think, an anecdote in my history here is going back about 10 years ago. I was just finishing grad school, and I was in one of my classes, my tumor biology class, and I remember that we one of the presentations was talking about cancer testing, and it was talking about the fact that there was a pathway, the JAK-STAT pathway, and how you did one PCR test and it was 7 to 14 days. Another PCR test was 14 days, and you had two more markers in there, right? So you had four targets in this pathway. Well, conservatively, that could be more than a month to get an answer. And in the time that I was a student to now, where I'm lucky enough to go back, my mentor in grad school, Jen Blancato at Georgetown University, she's invited me back to speak to the students in that same class. And I tell them, 10 years ago when I sat in your seat, this was the standard of practice, right? This was how things were done. This was, even in research at times, you'd have to do this type of PCR approach. But now with next-gen sequencing, with the parallelization of the technology, you could do hundreds or even thousands of whole genes at the same time. And you can get an answer, instead of doing sequential testing, you can get an answer potentially in 2 weeks, as quickly as 2 weeks, for hundreds of genes at the same time. And so when I look at that, right, where we've been in the 10 years since I've been a student, and I look at where we're going, we see there's such an exciting amount of development in single cell technology, right? Where, instead of looking at a tumor which is essentially a collection of cells, but tumors are not all the same. And so often, there are differences in tumors in the position, on where the tumor sits, and the genomic differences in that actually can lead to differences in how it behaves. So looking at single cells allows you to look at the differences individually of say, something like a tumor, and be able to understand the genomic differences and be able to potentially treat them or attack them, with therapeutics if there's something developed for that. Liquid biopsies, which, I spoke about, and QIAGEN has been at the forefront of developing technologies for isolation. I mean, that's obviously, cutting edge as people are looking to do, prophylactic testing ahead of time for potential cancer screening. We have spatial sequencing, which I think is super interesting because it's actually kind of a mix of anatomic pathology and in situ molecular testing, right? It combines those two things. So instead of looking at a pathology slide to see, fluorescence to see if there's a protein, you're actually able to take the same idea and look at the RNA expression profile of an individual area, right? So it's not just the proteins, it's actually molecular and potentially with protein. So those are all exciting areas, I think, generally where we see next-gen sequencing going, because all of that stuff leads to next-gen sequencing. At QIAGEN, though, one of the areas that we think is incredibly important that we've built out is multimodality, or what's called multi-omics, right? And we've built a portfolio around this to look at the DNA and RNA that's extracted from the same sample. And so, with our QIAGEN, with our sample prep kits, you're able to get nucleic acid, a DNA and RNA at the same time. So I can take one sample, I can take nanograms of input, and now I can do a comprehensive genomic profiling test, right? I can look at DNA mutations. I can look for RNA markers such as gene fusions. All of this can be done from nanograms of input. And when I talked about what it was 10 years ago, 10 years ago to do DNA and RNA at the same time would cost you $25,000 to $30,000. And now that you can do it, for hundreds of thousands of dollars, including the sequencing costs, right? So I think what you can expect in the future is to see more of that type of innovation as the costs decrease and the ability to expand and do, multiple omics at the same time. I think it's a really exciting technology that I believe, has a long future in whether in medical genomics and then research genomics as well.
Well, give us a website, if you would, where we can go and learn more about QIAGEN and the developments that are happening there.
Yeah, so if you go to www.QIAGEN.com and you type in next-gen sequencing, it'll take you to a page that has a comprehensive information about all of our portfolios. As I said, when I joined here in 2015, we had a handful of products, but since then we've built this comprehensive portfolio around DNA and RNA and epigenomics, infectious disease, right, including SARS-CoV-2 detection. All of this stuff matches up really nicely with our sample prep technologies that we hit that 3 billion mark with. So, if you come to QIAGEN and you're looking for isolation, or if you're looking for next-gen sequencing and you put next-gen sequencing in, you'll find all the information that you need to be able to work on, a cancer sample, a research sample, a COVID sample, or whatever it is that you really need.
Well, I appreciate you joining us here on the program this morning. It's been a pleasure, and hopefully you'll return in the future. Thank you.
Thank you very much, Neil.
You've been listening to Health Professional Radio. I'm your host, Neil Howard, in conversation with Mr. Brian Dugan. Audio copies of this program are available at hpr.fm and healthprofessionalradio.com.au. You can also subscribe to the podcast on iTunes, listen in, download it, SoundCloud, and be sure and subscribe to our YouTube channel at youtube.com, Health Professional Radio.